Good day, everyone. So another genus that we will discuss for today is genus Francisella. So what are the characteristics of this genus? They are gram-negative cocobacillary rods, and they are poorly staining bacteria. They are bipolar, non-motile, capsulated. So most of the species have the capsule, which is known to be a virulence factor of the organism. They are strict aerobic bacteria, fastidious. So they require the addition of cysteine in blood agar for enhancement of their growth. So the glucose cysteine blood agar is used for the culture of this organism. So this figure shows the appearance of the Francisella infecting macrophage. So the bacteria appears here as blue and the macrophage is yellow under electron microscopy. And the second figure shows the Francisella appearance in gram staining using light microscope. This is the growth of Francisella tularensis on blood agar plate. Various media can be used for the culture and isolation of the organism, Francisella. For A, it is the buffered charcoal yeast extract used to culture the organism. For the B, this is the chocolate agar medium. Letter C is the ship blood agar, and cysteine is letter D, the cysteine heart agar. So the organism has no known growth on McConkey agar. It is not able to grow on McConkey agar. For the biochemical characteristics, it is catalase positive because it is an aerobic organism. Oxidase negative, facultative intracellular pathogen. So it can survive even when engulfed by the macrophages. Has high lipid content, which is possible that can add in the ability of the organism to cause disease among animals. And virulent isolates usually produce capsules or are capsulated. The usual habitat of this genus Francisella. So there are reservoir hosts involved, and this includes lagomorphs, rodents, birds, and deer. The organism is known to survive in the environment for up to 3 to 4 months. So they are known to be stable in the environment. The species are Francisella novicida, which is known to be a virulent, so non-pathogenic or a virulent to rabbits, white mice, and pigeons. Francisella tularensis is known to share antigen with Brucella abortus, which was discussed already, that is known to cause abortion in cattle. There are 3 other Francisella species. We have Noatunensis, Novicida, Philomiragia. Pathogens of fish and occasionally associated with human infection is Francisella philomiragia. So there are reservoir hosts that are involved, and there are 100 species of mammals that can be affected. There are ticks, flies, and mosquitoes that can also serve to be sources or carriers of infection or carrier of Francisella organism. There are 2 biovars of Francisella tularensis, biovar type A and biovar type B. Francisella tularensis subspecies tularensis is known to be the type A biovar, which is known to occur in North America. It causes the classical tularemia in animals and human. So Francisella tularensis is an important zoonotic pathogen, widespread in wildlife, endemic in rabbits, known to hydrolyze glycerol that produces citrulline uridase, associated with terrestrial. You say terrestrial, that are the animals that are found in the land or living in land. So terrestrial animal reservoir are associated with the type A of Francisella tularensis. Next, we have the type B, or Francisella tularensis subspecies holarctica. It is known to occur worldwide and was more commonly reported in North America and Asia, Eurasia. Less virulent organism compared with the type A. Reservoir hosts are voles, water rats, muskrats, and beavers. Commonly isolated from aquatic animals and water-associated infections like from swimming in river is known to be the acquisition of infection. So this table shows the distinguishing features of the type A and type B. Type A, which is Francisella tularensis, subspecies tularensis, and the type B is the subspecies holarctica. In terms of their pathogenicity, the type A is known to produce more virulent disease in animals and humans. So it is the cause of classical tularemia, while the type B produce less serious disease in animals and humans. The reservoirs are lagomorphs, rodents, galliform birds for the type A. For type B, water, mud, and aquatic animals. Capsule production, both are producing capsule. Sustain is required both for their growth. Citrulline uridase activity is known to be manifest or demonstrated by the type A, and acid production from glucose is for type A. For the epidemiology, the tick and deer fly, like the Chrysops ziscalis, are important vectors in North America. The role of other vectors like ticks, Dermacentor variabilis, Dermacentor andersoni, and Amblyomma americanum are important tick vectors. Tick species in which Francisella tularensis can be passed transtadially and transovarially. So these are some of the pictures of the ticks which are most common source of infection to people because of the tick bites. So they can be infective for life. These ticks can be infective for life, but they are the sources of infection for humans. And these are the Dermacentor species and the Amblyomma that we have mentioned. The mosquitoes and flies, they are infrequent vector of disease in humans and only infective for 14 days, which is in contrast with the lifetime infective stage that the ticks usually can transmit the Francisella to human. But among flies like the Chrysops discalis, or also known as the deer fly, the organism or the fly can only be infective for 14 days. So the disease that the organism Francisella tularensis is known to be tularemia, or other disease. Other name of the disease are deer fly fever, rabbit fever, O'Hara's disease. This condition is a septicemic disease affecting more than 10 species of wild and domestic animals, birds and fish, even humans, as we have mentioned. Disease is highly infectious to man and endemic in rabbits. The disease resembles bubonic plague, which is caused by another pathogen, Yersinia pestis. Reservoir hosts are wild animal species, especially rodents and ticks. Primary host is sheep, secondary hosts are dogs, cats, pigs, and horses. Transmission is via insect vectors, the bites of the arthropods, skin abrasions, inhalation of aerosols, via ingestion. So the organism is very invasive. It's known to be intracellular parasite, spread via the lymphatics and produce miliary whitish foci of necrosis in the liver, spleen, and lymph nodes. So the disease in domestic animal is common. Disease was reported in sheep, horses, and young pigs. Adult pigs and cattle are comparatively resistant. Dogs and cats may be infected and seroconvert without clinical signs of the disease. When you say seroconvert, they are known to produce antibodies. That's why they are not manifesting clinical signs of the disease. For the pathogenesis and pathogenicity, since the organism is a facultative intracellular pathogen, it can survive in macrophages but not in neutrophils. Organism enters macrophages via a unique system of pseudopod loops. The organism is known to inhibit phagosome-lysosome fusion and replicates in acidified phagosomes. Acidification is very important for the release of iron from transferrin, which is required by the organism for its growth. Lymphadenitis, either local or generalized, is a constant finding. That's why we mentioned that it resembles the bubonic plague caused by Yersinia pestis. When you say bubonic plague, there is inflammation of the lymph nodes. We called it as buboes. Septicemia, pale necrotic foci, which is present in superficial lymph nodes, and miliary lesions in the liver and spleen can be observed. There are areas also of consolidation in the lungs. Clinical signs are fever, depression, inappetence, stiffness, and septicemia. These are the lesions that can be seen in the liver. There are gray necrotic foci of lesions that can be seen in the liver of rabbits. This figure shows numerous pale focal necrosis less than 1 micrometer in diameter in the liver lobe, in the lung also, and there are presence of pale focal necrosis in the spleen. And this is found in cats. This one is from beaver liver. So the liver of the animal shows disseminated small pale focal necrosis as shown in the picture. How do we diagnose heavy tick infestation among severely ill animals? And we can submit different samples or specimen for laboratory testing, blood for serology. We can submit scrapings from ulcers, lymph node aspirates, and biopsy material or postmortem samples. Fluorescent antibody technique for identification of Francisella tularensis in tissues or exudates and from cultures. Isolation procedures must be carried out in a biohazard cabinet because the organism is dangerous to be handled. That's why isolation should be carried out in a biosafety cabinet or a biohazard cabinet. PCR or polymerase chain reaction in blood, isolation in embryonated egg or lab animal can be used. Culture and identification of the organism using glucose-sustained blood agar with addition of antibiotics. Plates are incubated aerobically at 37 degrees Celsius up to 7 days. And the following are identification criteria for the isolates. Small gray mucoid colonies surrounded by narrow zone of hemolysis in blood agar plate. Immunofluorescence can be used to confirm the identity of the pathogen. A slide agglutination test can be carried out using cultures using anti-serum specific for Francisella tularensis. Biochemical test for distinguishing type A or the subspecies tularensis from type B or the subspecies Holarctica strains can be used. How do we treat? We can give antibiotics like amikacin, streptomycin, imipenem, cilastatin, and fluoroquinolones. Control measures, number one is control of the ectoparasites, especially the ticks, which are known to be infective for life, and also the presence of flies and mosquitoes. Prevention is preventing the contamination of food and water with infected carcasses or excreta of wildlife species. Prevent dogs and cats from hunting wildlife species. So there is no vaccine available yet for Francisella tularensis infection. So this is an important zoonotic pathogen. So we will discuss the disease caused by this organism in humans. So the disease is known to be tularemia, although it has also other name like the rabbit fever or Harris disease, which were mentioned a while ago. This is a serious and potentially fatal infection in humans, which involves slow healing ulcer accompanied by lymphadenopathy or inflammation of the lymph nodes, or what we call as buboes, who are at risk to acquire the infection: hunters, trappers, veterinarians, laboratory workers. In humans, there is a vaccine available for protection. Mortality rate generally when untreated 5 to 15%, when treated 1 to 3%. Typhoidal tularemia is 2 to 3 times higher than other forms. The type A, which is subspecies tularensis, has higher case fatality. Type B produces only few deaths even without treatment. In humans, there is 3 to 15 days incubation period. 7 disease forms of tularemia exist or reported in humans, but all these forms started with the following signs like fever, sudden fever, chills, headache, and myalgia or muscle pain. So the first form of tularemia in humans is the ulceroglandular form. This is the most common form of tularemia in human, which is caused by an arthropod vector or can be acquired from handling of infected meat. Next is the glandular form. There is no ulcer, but there are one or more enlarged lymph nodes. This is the second most common form of tularemia in human. So these ulceroglandular and glandular are the most common form of disease caused by Francisella tularensis, subspecies tularensis, and holarctica, which accounts for 75 to 85% of naturally occurring tularemia cases in humans. So these are the appearance of the ulceroglandular form, which involves the presence of ulcer and inflammation of the lymph nodes, which are painful also. So these are the ulcerations in the skin. And then this one are tularemia lesions on the dorsal skin of the right hand in humans. Next, we have the oculoglandular. Oculo, since it involves the eye, mainly in the conjunctiva. So by contaminated fingers, contaminated materials flushed into the eye are the common routes of infection. Clinical signs are flu-like conjunctivitis with regional lymphadenopathy. So the severe form, it is a severe form that involves ulceration of the conjunctiva and presence of ocular discharge. Next is the oropharyngeal form, which involves ingestion of contaminated material, so consumption of undercooked meat or water or contaminated water. So it involves hand-to-mouth transmission. Clinical signs are pharyngitis, diarrhea, abdominal pain, vomiting, gastrointestinal bleeding, nausea, and there is pseudomembrane that may develop over the tonsils. Next, we have the typhoidal form. This is the most deadly or lethal form clinical signs, septicemia, without lymphadenopathy or ulcer. Systemic infection can develop from the oropharyngeal form of tularemia. Next, we have the pulmonary form. Via inhalation of aerosol, complications from other form is the pulmonary form. 10 to 15% of the ulceroglandular and 50% of the typhoidal cases can result in the pulmonary form of the disease. Lastly is the intestinal form, which involves these clinical signs: abdominal pain, vomiting, and diarrhea. So that's the last slide for the genus Francisella. Thank you, and if you have questions, you can send them via our group chat. Thank you and good day.